everyone. Welcome to a new session on informatics and introduction to bioinformatics. Here we discuss next generation sequencing. The evolution of genomic science. DNA sequencing has come a long way since the days of two-dimensional chromatography in the 1970s. From 1977, with the arrival of capillary electrophoresis-based sequencing, researchers were able to sequence the full genome of any species in an efficient manner. After a decade, the first automated CE-based sequencing instruments called AB370 and AB3730XL were introduced, and it was the primary workhorses for the NIH-led and Celera-led human genome projects. Then, the genome analyzer was introduced to the research field on 2005 that helps to sequence 84 kilobase to 1 gigabase per run. Then, the parallel sequencing technique launched the next generation in genomic science. From that point forward, the next generation sequencing or NGS has used widely in the field of genomic research, and the data produced from NGS has increased every year. In 2005, the amount of data produced in a single run was 1 gigabase, and that is increased to 1.8 terabases of data by 2014. Beyond the massive increase in data output, the introduction of NGS helps the scientists to think about genetic information. The $1,000 genome establishes the foundation for personalized genomic medicine. It is now possible for the researchers to analyze thousands to tens of thousands of samples in a single year. The basics of next generation sequencing. Next generation sequencing, also called second generation sequencing, has similar properties to Sanger sequencing, but is with much higher throughput. The completion of the first drafts of human genome sequence was in the early stage of 21st century. In the ensuing decade, surprisingly new technologies for DNA sequencing have been developed. The highest throughput sequencers can generate over 20,000 times as much data in a single run as those used in the Human Genome Project. But even by 2015, a number of next generation sequencing approaches have come and gone, falling by the wayside and leaving only a very few real genome sequencing contenders left. The Roche technology used for Dr. Watson's genome was the first NGS technology to market, with the now long deceased GS20, so named because it was a genome sequencer capable of producing a massive 20 million bases of data. Compared with the most popular of capillary sequencers, Applied Biosystems 3730XL, it was a monster, producing over 250 times as much data in a single run. Its latest version, the GS-FLX, increased this output another 35-fold to 700 million bases, but even this was not enough. Roche announced in 2013 that they are shutting down its 454 sequencing division and that it would discontinue support in 2016. In the meantime, the company had other sequencing goals in sight. They attempted a failed hostile takeover of industry leader Illumina in 2012 and forged an alliance with the nanopore sequencing pioneer Pacific Biosciences in 2013. The concept behind next generation sequencing is that the DNA polymerase catalyzes the incorporation of fluorescently labeled deoxyribonucleotide triphosphates or dNTPs into a DNA template strand during sequential cycles of DNA synthesis. During each cycle, at the point of incorporation, the nucleotides are identified by fluorophore excitation. In next generation sequencing process, millions of fragments are sequenced in a parallel fashion. Illumina sequencing by synthesis is the most widely adopted one in the industry and delivers the highest accuracy without any error. The main advantages of next generation sequencing over other methods are that it gives simple, scalable and high yield data with low cost and fast run. The 4 common steps in next generation sequencing are library preparation, cluster generation, sequencing and data analysis. Now let us discuss each of these steps one by one. Library preparation. Random fragmentation of the DNA or cDNA sample is used for the library preparation, followed by a 5' and 3' adapter ligation. Also, the process called tagmentation helps to combine the fragmentation and ligation together and increases the efficiency of library preparation. The adapter ligated fragments are then PCR amplified and gel purified. First, DNA is fragmented to the optimal length determined by the downstream platform to get homogeneous blunt-ended fragments. End repair is performed to make sure that each molecule is free of overhangs and contains 5' phosphate and 3' hydroxyl groups. Libraries to be used in blunt-ended adapter ligation and can be used directly in the ligation step. For the libraries of Illumina and 454 platform, incorporation of a non-templated deoxyadenosine 5' monophosphate or DAMP onto the 3' end of blunted DNA fragments, a process known as DA tailing, is necessary. DA tails prevent concatemer formation during downstream ligation steps and enable DNA fragments to be ligated to adapters with complementary DT overhangs. The desired adapter ligated DNA size can be selected via gel electrophoresis before amplification by the polymerase chain reaction PCR. The graphical representation of library preparation has been illustrated in the following figure. Let us now discuss about the second step, cluster generation. Each fragment is captured on a lawn of surface bound oligos complementary to the library adapters and then amplified into the distinct through bridge amplification. In the process of cluster generation, libraries are denatured, diluted and distributed on the 2-dimensional surface of the flow cell and then amplified. While shorter products amplify more efficiently than longer products, longer library inserts larger and more diffuse clusters than short inserts. After this step, the templates are all ready for sequencing. As shown in the figure, a fragment library is produced in which adapters are ligated to opposite ends of the fragments. Linkers that are covalently attached to the surface of the flow cell capture the library and a bridge is formed between these. Isothermal amplification is then initiated from the double-stranded ends of the bridge. Displacement and re-annealing to the surface and amplification of the locally bound sequence thus forms a cluster of clonally expanded sequences. The third step is sequencing. There are different approaches for sequencing. For example, Illumina SBS technology utilizes reversible terminator based method that detects single bases as they are incorporated into DNA template strands. The last step involved is data analysis. The newly identified sequence rates are then aligned to a reference genome. Then the alignment is performed and is possible through different approaches such as single nucleotide polymorphism, SNP, or insertion deletion identification, read counting for RNA methods, phylogenetic or metagenomic analysis and more. Next generation sequencing methods. Next generation sequencing platforms provides variety of methods, helps scientists to ask virtually any kind of question about the genome, transcriptome or epigenome of any organism. Sequencing methods depends on the way how the DNA or RNA samples are obtained. For example, organism, tissue type, normal versus affected, experimental conditions. Also, the data analysis options used. After the sequencing, libraries are prepared. The actual sequencing stage remains fundamentally the same regardless of the method. Genomics, whole genome sequencing. The composition of first human genome was completed in 2003 as part of the Human Genome Project. During whole genome sequencing, the identity of 3 billion nucleotides that compose the human genome is determined from the prepared DNA sample. Today, sequencing technology is much more efficient and a human genome can be sequenced within few days for under $10,000. The whole genome sequencing is mainly used to create personalized plans to treat disease, not only based on the mutant gene, but also other related genes from the patient's genome. For example, genotyping of cancer cells and identifying the dysregulated genes allows physicians to select the best chemotherapy and potentially expose the patient to less toxic treatment. New candidate genes can be identified through whole genome sequencing, as the traditional methods concentrate only at the troublemaker genes. The steps in whole genome sequencing are shown in the following figure. In this procedure of whole genome sequencing, as you can see in the figure, a large DNA molecule is sheared into smaller fragments. The individual fragments are then sequenced. The fragments obtained are then assembled for overlapping sequences. Finally, the sequence is assembled. Exome sequencing. The most widely used sequencing method is exome sequencing. Exomes contains the majority of the known disease causing variants, even though it represents less than 2% of the human genome. Exome sequencing is a cost-effective alternative for the whole genome sequencing. In exome sequencing, only the protein coding part of the genome is captured and sequenced. Exome sequencing has wide range of application in population genetics, disease genetics and cancer genetics. A simple workflow of exome sequencing is shown in the figure. As you can see in the figure, in the procedure of exome sequencing, the protein coding region or exonic region is cleaved from the double-stranded DNA and suitable linkers are attached. They are then hybridized on capture array designed to target only the exons. De novo sequencing. De novo sequencing is a sequencing of novel genome where there is no reference genome available for alignment. De novo sequencing is usually performed by assembling the individual sequences to longer contiguous sequences called contigs, or by ordering the contigs correctly in the absence of the reference sequence. The quality of the de novo sequence data depends on the size and continuity of the contigs or the number of gaps in the data. Historically, capillary electrophoresis sequencers are used for the de novo sequencing. With its long read lengths and high accuracy, capillary electrophoresis based sequencing made overlap consensus assembly the gold standard technology for de novo projects. However, recently the high throughput capabilities of massively parallel sequencing and the development of short read assemblers helps to reduce the time and cost significantly associated with sequencing an entire genome. The advantages of de novo sequencing are: it is capable to generate accurate reference sequences even for complex genomes, provides useful information for mapping genomes of novel organisms or finishing genomes of known organisms, avoid repetitive and similar regions, helps to identify the rearrangements such as deletions, inversions or translocations. Here on the screen you can see a figure describing de novo assembly. In the method of de novo sequencing, the initial generation of primary genetic sequence of a particular organism is called de novo sequencing. A detailed genetic analysis of any organism is possible only after de novo sequencing. De novo sequencing is accomplished by assembling individual sequence reads into longer contiguous sequences or contigs or correctly ordered contigs in the absence of a reference sequence. Targeted sequencing. In targeted sequencing, a region of the genome or a subset of genes are isolated and sequenced. Targeted sequencing allows researchers to focus time, expenses and data analysis on specific areas of interest and enable sequencing at much higher coverage levels. If we sequence a coverage level of 30x to 50x per genome through a typical sequencing, the targeted sequencing provides us with a coverage of 500x to 1000x or higher. This higher coverage allows researchers to identify variants that are too rare or expensive to identify with whole genome sequencing or capillary electrophoresis based sequencing. The panels used for this sequencing can be purchased with a pre-selected and fixed content or can be custom designed. A wide variety of targeted sequencing library prep kits are available, especially kits including probe sets focused on particular areas of interest like cancer, cardiomyopathy, autism, etc. But the advantage with the custom design is that the researchers can target the genome regions that are relevant to their research interest. So, the custom targeted sequencing is ideal for analyzing genes that are involved in specific pathways or for the follow-up studies from the Genome Wide Association Studies GWAS and Whole Genome Sequencing Studies WGS. The targeted sequencing is of two types: target enrichment and amplicon generation sequencing. In target enrichment, specific regions of interest are captured to biotinylated probes by hybridization and then isolated using magnetic pull down. The target enrichment helps to capture regions with 20 kb to 62 MB depending on the library prep kit parameters. But the amplicon targeted sequencing involves the amplification and purification of the specific regions of interest using the highly multiplexed PCR oligosets. Using amplicon targeted sequencing, researchers can sequence 26 to 1536 targets at a time, 150 base pairs to 1.5 kb per target, depending on the library prep kit used. In this case, the multiplexed PCR provides a wide range of application for the discovery, validation and screening of the genetic variants. Amplicon targeted sequencing is very useful for the identification of somatic mutations in the case of complex samples like cancerous tumors mixed with germline DNA. Also, it has a wide range of application in the 16S rRNA bacterial sequencing across multiple species and in the area of phylogeny and taxonomy studies. The workflow of target enrichment and amplicon generation is described in the figures shown on the screen. As you can see in the figure, library fragments are first prepared, then oligos are added to block the sites. The targets are then hybridized to capture the probes. Then magnetic streptavidin beads are added to bind to the probes. The targets are captured with magnet and washed the target sequences away. Transcriptomics. The library preparation for RNA sequencing usually begins with a total RNA sample preparation and then followed by a ribosome removal step. The total RNA sample is then converted to cDNA before the new generation sequencing library preparation. RNA sequencing is mainly focused on mRNA, small RNA, non-coding RNA and microRNAs, which can be achieved by including steps like isolation, enrichment, etc. As you can see in the figure, the cDNA is transcribed from the total RNA. A pair of oligonucleotide probes designed to target and capture the regions of interest are used, followed by next generation sequencing. Total RNA and mRNA sequencing. Transcriptome sequencing is a major advance in the area of gene expression studies, as it allows having the picture of whole transcriptome rather than a predefined subset of genes. The whole transcriptome sequencing is capable to provide an extensive transcriptional profile at a cellular level that intensifies the power of RNA discovery method. Similar to other sequencing methods, this method also allows the identification and quantification of both common and rare transcripts. The additional benefits with this approach are the aligning of sequencing reads across splice junctions, detection of isoforms, novel transcripts and gene fusions. Library preparation kits are available for both total RNA and mRNA sequencing methods with the support of precise detection of strand orientation. Targeted RNA sequencing. Targeted RNA sequencing is a method that helps to measure the transcripts of interest for the differential expression, allele-specific expression and identification of gene fusions, isoforms, SNPs and splice junctions. Targeted RNA sequencing is a powerful method that can be used for the investigation of specific interested pathways or for the validation of gene expression microarray or whole transcriptome sequencing results. This method can overcome the difficulties in the wide range of cellular RNA population by concentrating only on the genes of interest and getting a huge range of enriched sequencing coverage. This can lead us to more sensitive and effective drug discovery and qualification. The most interesting fact with this method is that this sequencing enables the assembly of even low expressed transcripts. This method can also reduce the reagent cost as it uses multiplex library preparation. Small RNA and non-coding RNA sequencing. Small RNA or non-coding RNA or microRNA are 16 to 25 base pair nucleotides in length that participate in important functions like regulation of gene expression, gene repressors and gene silencers. The study of microRNA is growing very fast as the importance of microRNA in the transcriptional and translational regulation is more evident. The method can be used to identify novel miRNAs and non-coding RNAs that can characterize the changes like single base resolution. This method also has the advantage that prior sequence or secondary structure information is not needed for the analysis. Epigenomics. Epigenomics or epigenetics is a study of heritable changes in gene activity caused by mechanisms other than DNA sequence changes. Mechanisms of epigenetic activities include DNA methylation, small RNA mediated regulation, DNA protein interactions, histone modification and more. The epigenomics community was among the first to capitalize on the NGS application and development. That is, in epigenomics, the NGS methods are combined with established methods to capture epigenetically modified genomic regions. A key advantage of next generation sequencing methods is their ability to provide a comprehensive and unbiased view of the epigenome. It also helps the investigators from content limited microarray platforms. Methylation sequencing. Cytosine methylation is a much focused study in epigenetics that contains specific areas of regulation such as promoters or heterochromatin. Cytosine methylation can make significant changes in the temporal and spatial gene expression and chromatin remodeling. Methylation sequencing has the advantage on other methods that it can be used to analyze the methylation state at the single nucleotide level. The widely used methylation sequencing methods are whole genome bisulfite sequencing or WGBS and reduced representation bisulfite sequencing RRBS. With WGBS sequencing, sodium bisulfite converts non-methylated cytosine to uracil, which are then converted to thymine in the sequence reads or data output. In RRBS sequencing, DNA is digested with the help of a restriction enzyme called MSP1, which is unaffected by methylation status. Fragments in the 100 to 150 base pair size range are isolated to enrich for CpG and promoter containing DNA regions. Sequencing libraries can be then constructed using the standard NGS protocols. Chip sequencing. The interactions between protein and DNA or protein and RNA hold a significant impact on most of the biological processes and disease states. These interactions can be studied using combined approach of NGS and chromatin immunoprecipitation assays. Chip sequencing can vary according to the species, tissue and experimental conditions. This method is widely used for the identification of the genome-wide DNA binding sites for transcription factors and other proteins. Chip sequencing begins with the chromatin immunoprecipitation step using a specific antibody. Then, the chip DNA fragments are sequenced simultaneously using a genome sequencer. A single sequencing run can scan for genome-wide associations with high resolution, meaning that features can be located precisely on the chromosome. Here on the screen is a figure describing the workflow of chip sequencing. In the workflow of chip sequencing, the protein is first cross-linked to the DNA. The DNA strands are then sheared by sonicating. The cell lysate is then treated with bead-attached antibodies to immunoprecipitate the target protein. The unlinked proteins are removed and the DNA is further purified, sequenced, and the genome is mapped. Ribosome profiling. Ribosome profiling is a method based on deep sequencing of ribosome protected mRNA fragments. The purification and sequencing of these fragments are capable to provide a detailed picture of all the ribosomes that are active in a cell at a certain time point. This helps to identify the proteins that are getting translated in a cell and can be used in studies like translation control, gene expression measurement, determining the rate of protein synthesis, and prediction of protein abundance. Ribosome profiling enables systematic monitoring of cellular translation process and prediction of protein abundance. Determining what regions of a transcript are being translated can help define the proteome of complex organisms. With next generation sequencing, ribosome profiling allows detailed and accurate in vivo analysis of protein production. However, like every methods, next generation sequencing also has its own disadvantages. One of the major drawbacks that limits the use of NGS, especially in de novo sequencing, is the short sequencing read lengths that provide. A new technology which combines the massive throughput of the next generation sequencers with the long read lengths achieved by electrophoresis based Sanger sequencing would enable rapid, high quality production of de novo genome sequences. Also, setting up the required infrastructure such as computer capacity and storage and experts to comprehensively analyze and interpret the subsequent data. In addition, the volume of data needs to be managed skillfully to extract the clinically important information in a clear and robust interface. Let us now summarize what we have learnt in this session. In the past decade, many intractable questions are answered using next generation sequencing methods. New platforms for these methods are being developed constantly. As technology progresses, the main goal will be to fill in the sequencing space with platforms that can produce longer reads and more reads per run. The microbiome research area has developed tremendously from the advent of next generation sequencing technology. Currently, Illumina, which offers the highest throughput and lowest per base cost, is the leading NGS platform. However, next generation sequencing methods has its own disadvantages, in particular with the data storage and processing. Even though there are many databases available to deposit the data for public use and a wide variety of computational tools and software packages have been developed to deal with data from next generation sequencing data. In the coming years, the amount of sequencing data for different human genomes will increase enormously. With these genome data, confidentiality becomes an important factor. Ethical issues will definitely emerge with the communalization of personal genomes, and these issues urgently needs to be addressed. In addition, more efficient approaches to data storage and analysis are needed to keep up with the increasing speed of data production. Here are a few questions for you to work out. 1. What are the main benefits of next generation sequencing? 2. What is Solexa sequencing? 3. What is a contig in terms of sequencing? 4. What are the applications and limitations of next generation sequencing? 5. Explain the difference between whole genome sequencing and chip sequencing. Here I can suggest some books for your further studies. Statistical Analysis of Next Generation Sequencing Data, written by Datta S. and Nettleton D., published by Springer International Publishing House, 2014. Next Generation Sequencing Technologies and Challenges in Sequence Assembly, written by Sarah L. Metwally M., Auda O. and Helmi M., published by Springer, New York, 2014. Next Generation Sequencing in Cancer Research, Volume 1, Decoding the Cancer Genome, written by Wu W. and Chowdhury H., published by Springer, New York, 2013. Next Generation Sequencing, Translation to Clinical Diagnostics, written by Wong L., published by Springer, New York, 2013. High Throughput Next Generation Sequencing, Methods and Applications, written by Kwon Y. and Rick S., published by Humana Press, 2011. Challenges and Opportunities of Next Generation Sequencing for Biomedical Research, written by Rees M., published by Academic Press, 2012. Thank you for watching this program. Let's meet in the next session with a new topic. Until then, bye.